Hey class, today we're going to talk about the respiratory system and its regulation. So our goal is to basically just understand breathing. What causes ventilation or inspiration and expiration to occur. We're going to talk about gas exchange, the transport of O2, CO2 throughout the blood, and how with exercise, there's this hemoglobin saturation curve and it'll shift kind of right or left. And we'll talk about it shifting as we exercise, which helps to release oxygen so our muscles can use that. Take it down to the mitochondria, and then we'll go into a little bit of the neural control of the pulmonary ventilation. So, purpose of the respiratory system is to carry oxygen and remove CO2. Remember, we have pulmonary capillaries, then we have capillaries down at the skeletal muscle, and that's where diffusion or gas exchange occurs. This is carried out by 4 processes, so the inhalation of O2, removal of CO2 through pulmonary ventilation, diffusion, and then the transport of gases via the blood, and then the capillary diffusion. So the process of moving air into and out of the lungs, we have this transport zone and then exchange zone. Think of it as a transport zone, kind of the upper area where basically we're just kind of warming, humidifying, kind of clearing out some of the bad stuff within the air that could possibly hurt the alveoli. And then the exchange zone where, where we get to the alveolar sacs and gas exchange occurs. So ventilation is the movement of air between the atmosphere and the alveoli, basically just breathing in oxygen and removing CO2. So it occurs through inspiration and expiration. Okay, so here's just kind of where air would start, go through the nasal cavity, hit the pharynx, larynx, then the trachea, then we'll start to hit the bronchioles, and then get down to the alveolar sacs where there's the pulmonary capillaries, and we have the exchange of O2 and CO2. So here's just to give you guys an idea. I mentioned kind of the first part or upper areas of our respiratory system being more, these are where no gas exchange occurs, so we call these conducting airways. And then we kind of have the respiratory bronchioles, whereas we hit the alveolar sacs, that's where we have gas exchange. So you can see it moves down from the trachea, bronchi, bronchioles, terminal bronchioles, respiratory bronchioles, alveolar ducts, and then the alveolar sacs. That's where the respiration or the gas diffusion actually occurs. So the bronchi and bronchioles, their whole function is to warm, humidify air. There is no gas exchange, and it also removes foreign material from the air that could damage the alveoli if they reach it. Then at the alveolar sacs, this is where gas exchange occurs. Okay, there's a nice smooth laminar flow and receives nutrients from the pulmonary circulation. So you guys will figure this one out. It's the alveoli. And then just a little bit about the structure of the lungs. So the lungs are suspended by pleural sacs. We have the parietal pleura that lines the thoracic wall, and then the visceral pleura, meaning pulmonary pleura, that attaches to the lungs. So then the lungs, as we breathe in and out, takes the shape of the rib cage. So inspiration, breathing in, is going to be an active process. The involved muscles, the diaphragm kind of flattens out. The external intercostals move the rib cage and sternum up and out. So the thoracic cavity expands in 3 dimensions. And then expiration, breathing out. Typically a passive process. We see our lung volume decrease, our intrapulmonary pressure increase, and air is forced out of the lungs. Diaphragm comes back up. External intercostals relax. But then we have an active breathing, which this would be, or active expiration, which would be with exercise. We see the internal intercostals pull the ribs down, lats, abdominal muscles, forcing the diaphragm back up and really kind of forcing air out of the lungs. And so with expiration, we have this respiratory pump bringing actually helps to bring blood back into the right atrium. So as the pressure increases, we see this venous compression squeezing, okay, and that would be with expiration. As the pressure decreases, we see this venous filling with inspiration. So this milking action changes pressures and assists right atrial filling. So as we breathe in, helps to compress, okay, and bring some blood flow back to the right atrium. So here's the muscles involved. Let you guys take a look. Don't need to necessarily remember the location of these muscles, but have an idea of the muscles involved in an active and passive process. So with inspiration, as lung volume increases, so as we inspire, right, we're going to see that the intrapulmonary pressure actually decreases, and this is Boyle's law. So think of the P's kind of being the exact same size as the lungs expand. Okay, think of the P as like an oxygen molecule. The oxygen molecule does not get any bigger. The lungs get bigger, meaning the pressure decreases. Excuse me. As we expire, the volume of the lungs decreases, meaning the pressure will increase. So there's an inverse relationship there. And here's to kind of give you guys, here's the resting positions of everything. We can see the intrapulmonic pressure, intrapulmonary pressure is the pressure within the lungs, pulmonary lungs. Okay, intrapleural, okay, that's between the parietal and visceral pleura. Then there's pressure there as well. So as we inspire, we see both pressures go down, right? Lungs expanded with inspiration, pressure goes down. And then as we breathe out, okay, expire, we see the intrapleural pressure go back kind of resting, and then the intrapulmonic pressure increase above standard atmospheric pressure, which is going to drive us to breathe back in. So here's another graph to show you guys that both pressures go down and both pressures go up. Again, another image just to show you guys what's happening to the intrapulmonary and intrapleural pressure. So if we're measuring pulmonary volumes, okay, we have a pulmonary function test, which we start to measure tidal volume, vital capacity, residual volume, and total lung capacity. This helps us to determine respiratory diseases, okay, and whether the disease might be restrictive or obstructive. So total lung capacity is going to be sum of the vital capacity and residual volume. The vital capacity is the greatest amount of air that can be expired after maximal inspiration. Tidal volume is the amount of air entering and leaving the lungs with just a normal breath. The residual capacity is the volume of air remaining in the lungs after normal expiration, and the residual volume is the amount of air remaining in the lungs after maximal expiration. So we can see how that leads to total lung capacity with vital capacity and residual volume. So if we have restrictive lung disease, we're going to see everything go down. Okay, there's decreased compliance, our lungs are stiff, so they're not going to inflate as high, meaning, maximal inspiration, expiration is all going to be decreased, and we're going to have a difficult time breathing. Obstructive lung disease, we see it kind of inflate, meaning air comes in nicely, but it doesn't leave very easily, meaning it's going to kind of inflate, which is why we see that residual volume right here, and we see that total lung capacity. Both of those go up as well. So here's just to give you guys an idea of some restrictive and obstructive disorders. We see obstructive disorders could be something like COPD, asthma, and then restrictive disorders, if we are overweight, if we have scoliosis, some type of posture deficit, then obviously our lungs are not going to expand as big, and we're going to have a restrictive type of disorder. So how gas exchange occurs, okay, we see that the O2 comes to the alveolus via ventilation, and CO2 is removed from the alveolus during ventilation. We want to make sure that we have adequate perfusion, obviously enough blood, and then oxygen being removed, okay, and CO2 being breathed out. So as we look at gas exchange, okay, at rest, our lungs receive about 4 to 6 liters of blood. Okay, pulmonary diffusion, again, this is happening at the pulmonary capillaries of the alveoli. Okay, it replenishes blood oxygen supply and removes carbon dioxide from the blood. Here's kind of the oxygen cascade to give you guys an idea of the partial pressure of oxygen as it hits the lungs and the alveoli. So the pressure as it flows through and we hit the venous blood here, obviously we had gas exchange that occurs here, and then the pressure basically just keeps going down, down, and down. Again, going down because we are going to end up exchanging that oxygen and bringing deoxygenated blood back up to the lungs. So each different molecule has a different partial pressure. Basically, think of partial pressure as just kind of the amount of oxygen that we have flowing within our bloodstream. Okay, so we see that the partial pressure decreases, okay, as we start to hit the pulmonary artery, and then we'll see the pulmonary artery actually increase, okay, PCO2. Again, that's kind of, we have that gas exchange that occurs at the capillaries, so we're going to see as we're hitting the capillaries, PO2 will go down, PCO2 will go up. So here's just to show you guys between the pulmonary capillary and then the tissue capillary in terms of the amount of oxygen and CO2 that are carried within the blood. Again, just another image flowing through the left atrium here, down the left ventricle, and we see the PCO2, PO2 change. One goes up, the other goes down, again because gas exchange occurred. Again, just another graph so you guys take a look at that's in your book. So we transport oxygen. Over 98% of it is bound to hemoglobin. Okay, so we can call it oxyhemoglobin. Hemoglobin alone is deoxyhemoglobin, obviously without oxygen, but hemoglobin is going to carry oxygen throughout our blood. And now as we talk about unloading oxygen from hemoglobin, there's this saturation curve, and I want to make sure that we understand how we unload oxygen from hemoglobin. So there's an affinity. If the affinity is too high, so that means hemoglobin and oxygen like each other a lot, we're not going to want to release oxygen. But what's cool is as we start to exercise and we start to heat up, the affinity for those two starts to decrease. And what we end up actually seeing is that this curve will shift to the right, okay, meaning this oxygen is willing to dissociate easier from hemoglobin, and we can utilize that again because we see an increase in temperature, acidosis, PCO2, which happens with exercise, right? So it makes sense. We need oxygen for exercise. We see the affinity or them wanting to stay together decrease, so now we can take that oxygen a lot more easily. And then don't worry about the 2,3-DPG. Okay, that's just a metabolic byproduct of metabolism. So here's just that hemoglobin saturation curve. Again, you can see as our body temperature goes up, pH goes down, there's a decreased affinity, meaning we're going to unload oxygen, okay, to the skeletal muscle. So the AVO2 difference, I know I'm kind of going fast, but we're kind of flying. AVO2 difference is the arterial venous O2 difference, so the amount of oxygen within the arteries, the amount of oxygen carried within the veins. At rest, there's a high AVO2 difference, meaning there's not a huge change, right? We don't really need oxygen at rest. During heavy exercise, okay, it's a lot smaller because we have taken more oxygen up at the capillaries during exercise than at rest. As O2 hits the skeletal muscle, we know it's transported by hemoglobin in the blood and then to myoglobin as well. Don't worry too much about these curves. Here's just again the unloading, the saturation curve, and the PO2 curve combined, but we can see myoglobin and hemoglobin factors influencing. So the O2 content of the blood, again, is represented by the PO2 and hemoglobin percent saturation. This creates that gradient. Again, just kind of reiterating the fact that as pH goes down, temperature goes up, we see this unloading or promotion of unloading in the tissue. So now to end with carbon dioxide transport, okay, most of carbon dioxide is transported as bicarbonate ions or bound to hemoglobin, bound to the globin part. So 70% or so is carried as bicarbonate, okay, 7 to 10% in the plasma, the rest carried by hemoglobin. So what we end up seeing is CO2, okay, from the tissue binds with H2O, creates carbonic acid, and then dissociates into bicarbonate and hydrogen ions. These hydrogen ions can bind to hemoglobin and act as a buffer, again, because we don't want our pH to go down too low. So the rest is bound, like I just said, bound to hemoglobin. So as you can see, here's CO2. It's carried as a bicarbonate ion. This hydrogen ion, okay, is carried as hemoglobin, but we can also have CO2 transported to hemoglobin, which binds to the globin portion. The heme portion is what oxygen binds to. And then here's just a nice little picture for you guys to take a look at. And that will be today's lecture.